So we're moving into the fourth chapter of section 3, into cell functions. We're going to be dealing a lot with the membrane, so cartoon about membranes. I'm talking to you. You're so thick membranes sometimes. Do you have any idea what that is? These are old cartoons. It's a TV set with rabbit ears. Old cathode ray tube TV set. With basically the same technology as an electron microscope. Is that those big tubes for monitors and TV sets actually have an electron gun in it that shoots a beam that scans across the front of the tube and illuminates lines and spots to different degrees. First process, diffusion. Diffusion and heat work in similar ways. If you've got a whole bunch of stuff packed together and it's free to move, it will tend to spread out. If you've got a lot of heat in one place and it's free to be released, it spreads out. But we're going to talk specifically about diffusion of particles, although heat affects that. You take, again, lump of sugar. They were not common when I was a kid, but at least I've seen them. You used to actually, in a restaurant, there'd be a little bowl with little foil wrapped lumps of sugar, little cubes. And if you dropped it into a beaker of water, the outer part of the cube would get hydration shells. It would dissolve, but they would start to move through the water. And of course, there's a lot more movement away from the cube than there is of sugar back toward the cube. At this point, there's also diffusion of water into this place that the cube was occupying. As the cube loses sugar molecules, water moves in. So water is actually going from an area of high concentration into an area of low concentration. And that's diffusion. Movement of particles from where there's a bunch of them to where there's fewer of them, because there's more of them going in one direction than is going in the other direction. It is that simple. And it's based upon the fact that they're moving randomly in straight lines through a medium that allows them to move. Things move down a concentration gradient, high concentration to low concentration, high concentration to low concentration. Diffusion is a downhill movement in terms of concentrations, high to low. If it's going the other way, then you're not seeing diffusion. So when does it stop? Well, once that sugar is evenly distributed through the beaker, there's no area of high concentration or low concentration anymore. Everything is equal. So again, you've got an equilibrium. Now this starts to become a real issue, and the reason we led off with a picture about membranes is that cells have a barrier around them. and stuff moves through the barrier, or it doesn't. But when it moves through the barrier, one of the ways that it moves is by diffusion. It moves from an area of high concentration to an area of low concentration. And that's how a lot of stuff gets in and out of the cells. What can and cannot move through a barrier is related to the idea of permeability. It would be if I could spell. What can go through? Your cell membranes are permeable to oxygen and carbon dioxide. It just goes right through the membrane. It's not permeable to a tremendous number of large molecules. It's not that permeable to water. It's a little permeable to water. And so there are levels. And it's all comparison within a narrow parameters. Like, you know, this wall behind me is impermeable to most things, although I might be able to shoot a bullet through it, so it might be permeable to bullets. But it's pretty fair to say it's an impermeable barrier, except there's a door over here. And the door can change the permeability of the barrier by opening a hole in it. So you have impermeable, you have semi-permeable, some stuff can go through, some stuff can't. And in the case of living membranes, you have what's called selectively permeable, which is what can go through and what can't go through changes over time. Is that there's a certain amount of selection in the living system to go, okay, we need some of that, so let that in. Nope, that's enough, keep it out. You can be selectively permeable to some materials, to some particles. Also, one thing I want to mention, once the particles are mixed in and free to move, they are all moving more or less independent of one another. So, like when we do our diffusion lab, we use a salt water solution. And it's easy to say a salt water solution, but on a particle level, you got water, that's one type of particle moving. You have sodium ions, that's another type of particle that's moving, and you have chloride ions. Now the thing about sodium chloride is that when you put salt into water, then because it's coming out of an NaCl molecule, the concentration of sodium and chloride is equal. And under most circumstances, you don't just add straight up sodium or straight up chloride to things. So it tends in a lot of situations to stay kind of, you know, where the sodium's going, the chloride's going too. But they're no longer bound together. They're now moving independent of one another. A flux. Basically a fancy word of saying a flow. Net movement from one place to another. And it moves from, you know, high to low. A flux moves downhill. How quickly does it move toward equilibrium? That's affected by four parameters, two of which membranes can have some impact on. If you're talking about moving things through a membrane, Raise and lower temperature changes the speed the particles are moving at. Warmer, more particles are moving faster, then the flow is going to be faster. But in most living systems, there isn't going to be much of a control over temperature. You know, yes, we generate a set temperature in our bodies, but we don't warm ourselves up in order to move stuff faster. Particle size. It is something you need to be aware of, that if say you have a pore and, you know, 5 different things can move through the pore, the smallest of those 5 things is going to be able to go through faster because it's going to be able to squeeze more through at any given second. But if you're looking at one particular type of particle moving, it's got a size. There's not a lot you can do about that. Surface. It's coming through a membrane. How much membrane is there? Well, that is something that a cell can affect. It can extend membrane, it can stretch membrane. Microvilli, remember, is a way to increase membrane in a given area. So surface is something that you do see being altered in terms of membrane permeability. And then concentration gradient. Remember dynamic equilibrium, where the chemistry rate is going, you know, this way and then it's coming back. And one of the ways to keep it going that way is to take the products and do something with them or move them somewhere, so the amount of products doesn't build up to the point where it starts to come back. You can do the same thing with particles moving through a barrier. Oxygen coming into a cell moves by diffusion because the oxygen in the cell is being combined with hydrogen in respiration processes and it's becoming water. So there isn't a lot of oxygen coming back out. There is carbon dioxide coming back out because it's being produced by the same respiration processes, and there's less carbon dioxide out here, so you get a net flow. That's basically how breathing carbon dioxide kills you, is that eventually it doesn't, the concentration outside of the cells builds up to the point where you no longer are losing it, and then it does build up and mess with the respiration processes on the inside of the cells. It's not as, you know, carbon monoxide actually blocks oxygen from being carried by your hemoglobin. That is directly poisonous. Carbon dioxide is kind of indirectly poisonous, sort of, kind of. Water becomes a really big deal. In living things, the particles are dissolved in water. But the water itself can diffuse. Or diffuse, if I could pronounce that right. So water balance becomes a really big deal. Life evolved in the oceans. The chemistry inside those living cells developed in water balance with their surroundings, so that the amount of water in the cell was isotonic to the amount of water around the cells. Why? Well, then there's no net movement of water from a high concentration to a low concentration or a high concentration to a low concentration. So it was one less thing to have to do. If you're, okay, tell you how I learned this. This is opposite to what it makes sense in my head. To me, hypertonic sounds like more water, but it actually means less water. If you're in a hypertonic environment, then there's less water around the living system than is inside. If you're, or the membrane, if you're doing a non-living system. If you're in a hypotonic, then there's more water around. Now your tissues are very close to being isotonic to ocean water. We're a little bit more dilute than ocean water because our ancestors evolved in freshwater systems, and although they couldn't become isotonic to fresh water, they got a little bit more dilute over time, and we are kind of the descendants of that. But still, our cells are not 100% water. And so your blood cells have a certain osmolarity that matches the osmolarity of your plasma. No net flow. You can actually kill yourself by drinking too much water. If your kidneys are very good at keeping your plasma in water balance to your tissues so that you don't get this net flow. But if you overload the system by drinking lots and lots and lots of water, then your blood gets too dilute. Water goes by osmosis, diffusion from high concentration in the plasma into the low concentration of your tissues, especially your brain, because that's going to show the effects quicker. And it can, make you crazy and eventually kill you. Okay, here's the situation where this is in a solution that would match the osmolarity of plasma. You could do that mixing anything into the water, but here there's a water balance. What's going in and what's coming out is in equilibrium, same movement in and out. Now here is what our ancestors faced getting into fresh water. They were in a hypotonic solution, much more water around them than was inside their cells, because flowing off of rainwater flowing off picks up very little in the way of particles, as it is very close to 100% water. And you put a blood cell into that solution, more water is going in than is coming out. It will swell it up like a balloon and pop it. In fact, the stuff that we learn about membranes, a lot of that early work was done with red blood cells. It's neat, red blood cells don't have, I won't say any, but they don't have much in a way of organelles in them. They're basically a bag of hemoglobin, no nucleus, but a membrane. And if you take a bunch of red blood cells, you filter them out, and you put them into just a beaker of distilled water, they will swell up and pop. The hemoglobin inside mixes into the distilled water. You can run those through a filter and capture essentially just the membranes. And that gives you something to figure out what the chemistry of the membranes are. So the early work done on membranes was done basically by doing this. Now, if you put these cells into water that's very, very salty, then there's more water inside the cells to come out than is out in this salty solution to go in, and the cells will shrink up. When we do the lab, that should sound familiar. That should echo in your head. Thanks. If water moves into a system and then pushes back against the barrier, it generates osmotic pressure. That's what swells the red blood cells up to the point where they pop, is that you've got a whole bunch of water inside, and the way it goes in and out, it's still a barrier, is that if you have a screen up in a heavy wind, you'll see the screen belling out from the wind pushing against it. Yeah, the air can go through it, but it's also hitting the screen. And that's what happens when the cell swells up, is that the water is flowing in and trying to get back out, except there's too much of it. And a lot of it's pushing on the membrane itself. It's swelling the membrane up to the point where the membrane can't handle the pressure and it pops. There's a couple of specific applications of this. You have a plant with its roots down in the water, or down in the ground, sorry. It's in the water, groundwater. Rain goes into the dirt. It looks dirty, but it doesn't pick up a tremendous amount of particles. It's still very, very dilute water around these living roots. So there's more water on the outside of the roots than is on the inside. And so osmosis moves water into the roots. Doesn't need to do anything about it. It's a passive system. Now farther up above the ground, there's photosynthesis going on. The root pressure can push water into and up the plant. And on top of that, the plant's using the water for photosynthesis. So the concentration up here is dropping because the water is being converted into glucose. Well, some of the water is being converted into glucose. Oxygen's coming out, the hydrogen's going in and do stuff. We already talked about that. So the concentration gradient is sustained, is that there's always more water around the roots going in and up than is in the plant. The point, you hit a point though, where this water has weight. And so the root pressure has a limit of maybe as high as my thigh before the water moving up is just too heavy and it can't push against gravity anymore. But that's great for little plants. Little plants can get water up to the top of their plants with root pressure as long as they're fairly close to the ground. I know, I'm squeezing this in here. We talked about central vacuoles that push out on plant cells in small green plants and add to the stiffness of the plant. And if the plant hasn't had water for a while, they start to use some of that water for photosynthesis, and they lose their turgor pressure, the pressure of that balloon, and the cell and the plant gets wilty. Now they do that by putting particles into the central vacuole. Water flows in and pushes back out against the membrane. Something similar happens in contractile vacuoles, where water's flowing into these cells like amoebas that live in fresh water. They need to pump it back out. Well, it turns out water is notoriously difficult to actually grab and move in and of itself. You need to kind of set it up to move and then get rid of it. So what the edge of the contractile vacuole throws a whole bunch of particles into the contractile vacuole, and water follows because it's going from a low concentration or high concentration in the cell to a now low concentration in the contractile vacuole. And what happens is the membrane of the contractile vacuole very, very quickly pulls the particles out and then squeezes and squirts the now mostly fresh water back out. It's a bailing system. But of course, you can't just throw the particles in and get rid of them, or you'd be out of particles really quickly. So you throw particles in, osmosis fills up the contractile vacuole, you grab the particles and squirt the water out. That's one of the ways to adapt to fresh water. I'm not gonna write this down, you're gonna write this down. As marine animals from the salty oceans moved into freshwater systems, they had this issue of more water around us than is inside our cells. Osmosis is going to move water in. In the staging areas for movement onto land, flowing rivers and streams and tidal pools, rains into the tidal pools get dilute. Of course, freshwater outflow from a continent is very dilute. But you have this mixing area, estuaries, where you can kind of gradually evolve to the system. But how do you evolve to watery environments? First thing we just talked about, pumps. You can take the water as it comes in and pump it back out. That works. Freshwater animals like fish actually do some of that with their kidneys, but they don't do much of it with their kidneys. Why not? There's another thing that you can do is you can waterproof a significant part of your outer surface. You can make your outer surface impermeable to water, and then you don't have to worry about it coming in. And that's what our ancestors, the bony fish, did when they moved into freshwater systems, is they waterproofed most of their surfaces. Now, it turns out you can't waterproof them all. You can't get oxygen and carbon dioxide through a waterproof surface, so the gills, the breathing system, had to be open to water movement, and the digestive system had to be open to water movement. And so it's a combination of mostly waterproofing, and then your kidneys taking the dilution of your blood and getting rid of the water. So, pumping. If I were to give you a balloon, little balloon, blow this up until it pops. Most of you could do that. If I say, assuming this was empty, stick the balloon in there first and blow it up until it pops. You could blow it up, but once the balloon was in contact with the plastic bottle, I mean if this was empty, unless you've got extremely strong lungs, you're not getting that balloon to pop. Plants moving into fresh water didn't have the same danger that animals moving into fresh water did, because they have a casing around the cells. Same with funguses, same with a lot of bacteria. They have cell walls. And that cell wall is like that plastic bottle around the balloon that is the living cell. Osmosis moves water into the cell, gets it out to the cell wall, whatever casing it is. And now the only way for water to come in is when water bumps out. So you wind up with an equilibrium of flux because there's no more room for the inflow. Is water opens the thing up until it hits the cell wall, and then it can't make it any bigger. So you're not in equilibrium water-wise, but you're in equilibrium of flux, flow in and flow out. So that one doesn't really go on the list. I'm not really sure how it would go on the list. Okay, that's a good place to stop.